right, so today we're going to be talking a lot about chemistry, is that right?
Pretty much this is topic B for Micro 150.
All right, so what are some of the main learning outcomes that we're looking at today?
So this topic covers a couple of things. It's going to cover general chemistry and organic chemistry. So the idea is to get kind of an overview of both of them. We'll start with the basics from elements, atoms, compounds, and then we'll move on to specifically carbon to start building up much larger substances called macromolecules, which is really what we're going to be dealing with the rest of the class.
All right, so starting at the bottom, what are the main differences between matter and energy?
So when we define life, the idea is life is usually defined as something that is made up of matter and has energy. That doesn't really give us the whole picture. In reality, most textbooks now give us that they have more than that, called the properties of life. And those things like metabolism, growth, and structural similarities, we will cover those in separate lectures. So for right now, it's just kind of important to kind of discuss what matter and energy are, more or less.
All right, so getting into specifics, you got mass, energy. I know there's the two types of energy, and then mass, so that's the amount of matter in an object?
It's the content, how much is in it, and the good thing about it is that it's always constant no matter where in the universe you are. But that amount of matter or substance has or possesses energy, and depending on where you have that energy by location or how it's moving, it'll get things like being kinetic energy, so the energy of motion, versus potential energy, which is usually the energy of composition or position, and that's really the one we're dealing with in this class. So going into matter, we're going to discuss what things are made up specifically.
All right, so now we're kind of getting into periodic table stuff. You've got elements, there's isolated elements which are pure, then combined, so you can mix them like metals and things like that.
Correct. So we'll start with the pure ones, the ones that more or less cannot be broken down into more simple things. In other words, they lose their properties if they are broken down to more or smaller items. So the smallest you can get an element into is called an atom. And so the atom more or less represents the smallest amount of that specific element. The example I always give people is take any classic element you're familiar with. I use the example here of copper, in which it doesn't matter how much copper do you have, whether it's a ton of it, whether it's a bucket of it, whether it's a spoonful, or even just the tiniest amount that you can make it, which is an atom, it always has the same property. In other words, it cannot lose its properties, so it means it tastes like it looks like it behaves like it, and so on and so forth, as long as it's that same one element or that same one atom. So the rule is simple. An element in its smallest available quantity, which is an atom, always retain their properties. They always look like them, they behave like them, they react like them. So this is the smallest you can make something without losing its properties. Now, technically, you can make it smaller, but you have to apply more force and more energy to do so. And if you do that, you break the atom into subatomic particles, or smaller than atoms. And for that, we enter the two classic regions of an atom. This is what pretty much everybody learns in general chemistry. You have the center of it, or so-called the nucleus, and then the outer area behind it called the cloud. So we can kind of discuss these two really quick. This is the classic image that every textbook gives you, a core at the center that contains protons and neutrons, and the outside or outlying set of particles called the electrons kind of floating around in this little cloud.
So when we break up those individual pieces, those are the subatomic particles, right? So the specific, like the electrons, neutrons, and protons, those are subatomic particles.
Right. So we'll take first the core ones, and at the nucleus, there's two of them contained in there, which are the protons and the neutrons. And each one of them come with two things, two properties that we need to know: their charge, in terms of electricity, or their weight. And so the charge for the protons are always positive. The charge for the neutrons are always neutral. Their name kind of implies that. But both of these two particles weigh roughly about the same, which is referred to as an atomic mass unit, or AMU. Outside of that, then you have the cloud, which is where the electrons are usually kind of zooming around in all sorts of directions and kind of around the nucleus. And all of these guys possess a negative charge associated with them. But their weight is that they're so small that typically they're considered to be nearly weightless. And so most of the time in basic science, we just don't consider their weight. However, there are a couple of rule of thumb that we should know about these guys. In other words, how many protons, how many electrons, and how many neutrons does each possess. And that's what the periodic table is for. But just as a general rule of thumb, whenever you count the protons, you virtually have the same number of electrons and, roughly speaking, the same amount of neutrons. So a couple of examples that I can give you. Hydrogen, if you look at your periodic table, comes at the number 1, has 1 proton. By rule of thumb, should have 1 electron also, but it should have roughly the same amount of neutrons. And the difference between 0 and 1 is close enough. If we deal with the next element that we're going to be interested a lot in this class is going to be carbon. That comes with 6 protons, which means that by default it should also have 6 electrons and also roughly the same amount of neutrons. In this case, 6. Now the weight, though, again reminding you that the amount of weight that both protons and neutrons have are about 1 atomic mass unit, and electrons weigh nothing. When you do the math for hydrogen, you had the 1 of the proton and nothing else, ends up weighing roughly around 1. If you look at carbon, on the other hand, has 6 protons and 6 neutrons, each weighing about 1 each, so that means 6 times 1, 6, plus the other 6 gives you roughly a weight of about 12. The electrons, again, not really contributing to the weight.
All right, so getting into the periodic table, so that's something that a lot of people are familiar with, and I know it's kind of intimidating for some people. There's all the known elements up there, but it is organized in a certain way, right? So we got usually metals over on the left, gases on the right, things along that line, right?
Right, they are organized based on their properties. Now, we don't really need to know those properties because, again, this is not an absolute chemistry class, but it's kind of important to know how they're sorted so we can look them up. And the way they sort the table is fairly simple. They're organized by type, meaning certain amounts of properties. They're also organized by how many protons they have, so they go from 1 to about 120 currently. And they are also organized by columns behind how they behave. So some of them are really reactive, some of them pretty much do almost nothing, and so that's more or less the sorting of the periodic table. So for us, it's kind of important to know how to read the periodic table itself. So what are the things that we need to know about the periodic table are 4 main items, which are going to be how to detect its atomic number, which more or less means how many protons are in that atom, the name and the symbol, and then the weight, which really is the one we're going to do a little bit more work with. In other words, determining how much does it weigh by rounding off the number that you see in the periodic table. And the way that that's determined is pretty much by adding the number of protons, the number of neutrons in terms of their weight, reminding you that, again, the electrons don't weigh anything for our purposes. So if we use the example here of the periodic table, I can give you the example of hydrogen, that if you look at its atomic weight, it says 1.008. We kind of round that number up, or down in this case, and make it 1. If we look at carbon, for example, which says 12.011, we'd round that down to 12. Or if we looked a couple of units to the left, and we look at boron, for example, that says 10.811, that one would be rounded up to about 11. Now, what we want to know about these is, again, how many protons, how many neutrons are present. And so the rule of thumb is, again, number of protons, number of electrons are exactly the same, and the number of neutrons is roughly the same. So how do we determine that rough number of neutrons? Well, we do a little bit of subtraction and addition. You take the atomic weight that has been rounded, and usually you subtract the number of protons. So since hydrogen is 1.008, we round it up to 1, and subtract the number of protons that are present in there, which is also 1, and it gives us 0, meaning it has 0 neutrons. For carbon, which weighs rounded down to 12, subtract 6 protons that it says it has, it gives us also 6 possible neutrons that it contains. If you look at boron, since we're going to round it up to 11, its atomic number is 5, which means 5 protons. 11 minus 5 also gives us roughly around 6 neutrons. So as long as we can do that more or less math, that keeps us kind of safe in understanding the chemistry and the properties of the periodic table. So now what we want to do is learn how to combine them. Now that we understand more or less the basic units of chemistry, elements and atoms, how do we start building things out of them? Reminding you that elements, again, are the basic building blocks. They're the smallest amount without losing any properties. Now we can combine them with other atoms, different atoms, mind you, to start building things like molecules or compounds.
All right, so speaking of molecules, compounds, so first off, what is a compound?
All right, so the rule is very simple, but keep in mind there's a very specific definition to it. It requires 2 or more different elements or atoms, so that means 2 different sets of atoms on the periodic table, but that they have to be in a set proportion. What does that mean? more or less recipes. In other words, you have to have one of this, two of this, three of this, and that has to stay that exact amount. You can't change them, otherwise it changes the compound itself. So, in other words, the smallest amount we can make these compounds, more or less, we define them as a molecule. And that's where kind of the system kind of starts assembling itself. So what do we mean by this? You take an element that is already pure, you can't break it down into anything simple, you combine it with a different element altogether that is also pure, and you start building compounds. Of course, there are rules to build these, but as long as you follow more or less the rules of chemistry and the quantities, you get what's so-called a compound. And so the same rules that we talked about before, a ton of compound, a spoon of compound, a bucket of compound, all behave also in the same type of properties. And the smallest you can actually make those guys are called molecules. So you can have a comparison really quick. You can have lots of elements, but the smallest amount of an element becomes an atom. You can also have lots of a compound, and the smallest amount you can make a compound is called a molecule. So that's the relationship between those two in terms of size or quantity. But both of them follow the same rule. You can't make them any smaller without losing their properties. An additional rule that has associated with compounds and molecules is, again, recipes, proportions. You have to have a set amount of each. So in this case, let's kind of look at some of the examples of what a molecule can be. So again, reminding you that it's two different atoms interacting together, or two or more, I should say, and they are in the set proportion. So let's give you a quick example. A quick compound that most of us are familiar with is by combining two specific different elements in set proportions. So here we have carbon and hydrogen, and we're going to take one, in this case, atom of carbon and 4 atoms of hydrogen. And again, without kind of going into the detailed chemistry, we're going to form one compound called methane. Now, that's the compound methane, which we know is a gas and it's very flammable, and we actually use it in everyday life these days. But that amount of it, whether it's 1 gallon of it, whether it's 1 truckload of it, whether it's a spoon of it, it's referred to as a compound. Now, the smallest you can make that without it losing its properties of methane is called a molecule of methane. And so when you look at it, we'll kind of deal with this a little bit later, kind of its arrangement. You can see, again, it's set proportions. It's a recipe. 1 carbon, 4 hydrogens, that's the rule. It's the idea behind a set proportion. Now, that being said, it's kind of important to know a little bit more properties behind these compounds and elements. And one of the most common ones is talking about things called isotopes, which for us is kind of important in terms of biology because it helps us with understanding the age of certain things. Reminding you one more time that in terms of our atoms or our elements, these are pure substances. Now, every now and then, some of these atoms do actually vary in the amount of neutrons that they have. Again, you can't vary the protons because that would change the element. So if you went from hydrogen, which only has 1 proton, and you change the number of protons to 2, well, that became helium, so it stops being the same one. However, you can change the amount of neutrons. So hydrogen usually comes with 0, but you can actually give it another neutron or a second neutron, and it still remains as hydrogen. And so those variations in weight, now changing from hydrogen weighing 1, 2, or 3, is what we call an isotope. It's still the same element, still the same amount of protons, but its properties change just a little bit. Now we can talk about a different one. An example that we have here is phosphorus. So you can look it up on the table. And on the table here, phosphorus usually has 15 protons and 16 neutrons, but sometimes it can have a little bit more neutrons or a little bit less neutrons, going to 15 or 17, and the weight changes. But it's still phosphorus, and it still has the same amount of protons. Now, why are we learning this whole change in weight thing? Well, because most of these variations in weight usually want to go back to its original or most stable state. So hydrogen wants to go back to 0 neutrons, and phosphorus wants to go back to 16. And anytime it does that, this is called radioactivity. The energy that gets released by going back to those original states is called radioactivity. So we define radioactivity as kind of the energy that is emitted by isotopes. Now, not necessarily all isotopes end up being radioactive, but most of them have some degree of it. But it's kind of important to understand that the term radioactive doesn't necessarily mean that it's damaging or that it can cause you harm. Phosphorus can be a little dangerous in terms of radioactivity, but hydrogen in its two radioactive states, hydrogen 2 or 3, which are deuterium and tritium, do absolutely nothing to you. So they don't even pass through your skin, for example. However, there's ways to detect these, and pretty much this is what everybody's familiar with from most media. They think about things like Geiger counters, those little boxes that people are running around inside giant suits and is beeping at them. That's one of the tools that we have. And things like photographic film and X-ray film is a different way of detecting that. Now, why we want to understand the property of radioactivity for us, or something being radioactive, is that it helps us measure time. And so the amount of radioactivity something possesses as it's going back and forth tells us how old that particular element is. So going back to hydrogen as well as carbon in this case, they both have radioactive states, and they also take a certain amount of time to go back to their original state. Those times are very, very accurate and allows us to do things like carbon dating, which usually ends up telling us how old, for example, a tree is, or how old has a body been buried, or something like that. So this is an important property to know about our elements.
All right, so now we're getting into how elements react with each other, right?
Right, so this is the term that most people learn about, a chemical reaction, or just the word reaction itself.
All right, so chemical reactions. So when atoms combine, if they're able to react with each other, they'll actually rearrange, rearrange with each other, form a new combination, right?
Correct, and so what they're rearranging specifically was electrons. The atoms themselves don't trade in protons or anything like that, but they do like to trade electrons more than anything else, and they do it in several different ways. But what's unique about this rearrangement or combination is that it pretty much always involves some sort of energy exchange. Either energy has to go into the system or out of the system, or something thereof. Now, we're going to learn about this in terms of the basics of chemical reactions, simply to understand biology, not necessarily to understand the concept of a chemical reaction. So here we're going to be interested in how it exchanges the energy, so that we understand how animals and other living things, including bacteria and everything that might make us sick, exchange this energy to either grow, develop, divide, and so on and so forth.
All right, so we got about 6 different types of chemical reactions that we're going to get into, right? First one's acid-base.
Right, and so we'll talk about this one in a couple more slides, but like you mentioned, the 6 that we have, all of these kind of have importance in terms of how they affect biology. And what we're going to do here is kind of cover one by one as we go through the class. These are just the highlighted ones that we like to talk about in biology more than anything else. Clearly there's hundreds, if not thousands of these guys, but these are the ones that we consider to be important. For example, the dehydration one and the hydrolysis one will come a little bit later in the organic chemistry side. So instead of kind of covering them right now directly, what we'll do is cover them a little bit after as we get exposed to each of these topics.
So getting into chemical equations, this is another one of the things that can be kind of intimidating for people, but I don't think we get too in-depth in it in this class, right?
Right. Again, so to remind that this is just an introduction to the subject so that people kind of get a refresher. Here, the concept of a chemical equation is to remind people of how we write the reactions that are occurring. And here we're going to summarize it almost to basic math, if you will. And what you have, 1 + 1 = 2. But instead of using the equal sign, we use an arrow to indicate which direction the reaction is going. And there's various different forms of putting two things together, or breaking down one into multiple pieces, or converting them. Now, the example that I have written down here, not that it's even accurate, believe it or not, but the idea is to kind of indicate that you're taking one substance plus another substance and ultimately creating another. So the idea of 1 + 1 = 2 is being put there. And so that's really what we're highlighting. The idea that this is our shorthand way of writing things down, that way can people visually understand what's occurring inside a living system.
All right, so let's talk about bonds. So you were mentioning that when you get enough of an atom together, you could have like a bucket of copper, or down to the smallest piece, it's still the same atom. But bonds is talking about how they actually stick together, right?
Correct. So let's see more or less what we're interested in here is how they stay together. So how do they stay together? Usually the consensus is they're using some sort of force. The idea behind them is when they combine, when they undergo chemical reactions, is that their electrons are usually kind of rearranging themselves in some sort of way that is energetically favorable, or meaning they're more stable that way than they were originally apart. What's really neat about this is that even though it's a fairly complex process, we can actually draw this out by using what we call a chemical formula. chemical formula. In this case, there's two versions that we're going to be interested in. The first one being a molecular formula. And really what that means is it tells us just how much of each component. In other words, if you're talking about something as simple as water, it just tells us that there's two atoms of hydrogen, one atom of oxygen interacting together. That's really what the molecular one does. The other one that we usually hear about is the structural version of it. And this one gives us the arrangement in three-dimensional space. So how this actual compound, in this case also water, is distributed. In this case, you see that the water, the two hydrogens, are interacting directly with the oxygen. And this is as far as we're really interested in terms of formula. The idea is how many and more or less how it looks in space. So that's really where we're going to leave it. Now, how do they form is really what we're interested in. And again, reminding you that what it's using is usually those outer electrons in that cloud are the ones that are interacting and trying to stabilize. So what normally happens is there's this unique property of those outermost electrons, the ones that are outside or closest to the outside of the shell in terms of the cloud. Those guys are a little bit more reactive. And they kind of like to hang out in pairs most of the time, or at least in twos. And so if they're more or less hanging out by themselves, they'll seek out other electrons to find this kind of pair or stabilizing version of it. So what we're going to learn here are how do those pairs form and which kind of reactions occur and how do those bonds more or less kind of stabilize them. More or less, we're going to talk about three versions: the ionic bonds, covalent bonds, and hydrogen bonds, and each one of them has their unique process. When we start with these, the first ones we're talking about is the concept of an ion and eventually how those ionic bonds form. So here it's a very kind of simplistic version of that, in which you have two atoms kind of hanging out side by side, and what I'm going to more or less kind of highlight is that some of those electrons will usually shift their direction from one to the other by trying to create those pairs. Again, they don't like to usually hang out by themselves. The problem with this is that some elements or atoms lose those electrons, well, their charges change, and so some of these elements become positively charged while others become negatively charged as they lose or gain those electrons. And that's more or less what an ion is. It's usually the same atom, however, its changes in electrons now causes it to have charge, or more or less electricity. So our ions, in this case, are usually atoms that have lost or gained at least one electron. They can be more than that. And because they have now become charged, or they've become electric, they'll start attracting each other based on opposite charges. So positives and negatives will more or less attract. And what they'll seek out to do is these ionic bonds to neutralize those charges. So the equation that I had shown you earlier, the reaction that I had showed you, I told you was a little bit inaccurate. Now we see again sodium and chlorine in their ionic forms, so sodium and chloride. And because each one of them has a different amount of electrons, now sodium becomes a little bit more positively charged, chlorine becomes a little bit negatively charged, and those two opposite charges attract and come together. Other versions of them can occur in various different forms of the amounts of electrons, but the idea is bonds form as a way to stabilize those charges, so opposites attract here, as a way to neutralize those electric amounts of charge. Now, how are those bonds forming? Again, it is based on an attraction of the opposite charges, and that holds them together. Again, it's all about forces bringing things together. However, these forces are kind of weak, and most of the time, anytime you place them inside fluids, more than anything else, water, they break down really quick. So that same sodium chloride that we talked about earlier, if you pour it into water, both the sodium and the chloride ions immediately separate out into their original ionic forms. So this is kind of a weak one that we talk about most of the time. Now, in biology, the reason why we're interested in ionic bonds is because these substances create what we call electrolytes, substances that when in water conduct electricity. And as the human body and most living things, their functions are largely based on how they can transfer these electrons and more or less conducting electricity. So that's a very important point for biology. Now, if we want to go into some of the important versions of them, then you mentioned this earlier when we talked about the types of reactions that occurred. Some of them were called acids and bases. Well, here we're only going to take a very simplistic look at these compounds, and The idea for us is the definition is very, very basic. The idea that acids, anytime that they're inside water, part of their composition releases usually protons or hydrogen ions, whereas bases are slightly different in which they will release a hydroxyl ion to do so. And what's interesting about these reactions is that whenever you put acids and bases together, they form what we call salts. So anytime you put an acid or a base, regardless of their properties, they end up producing some sort of salt compound and usually as a result some water. And so we'll deal with those as we go into further classes a little bit later. Now, what we're going to want to do is also understand that salts, when they're present inside a body, whether by nutrients or in terms of physiology, these again are important simply because they form electrolytes. Anytime you place them inside water, they dissociate and they can conduct electricity. That's our biggest importance behind these guys right now. Now, the other reason behind why these are important is because of something called pH. And more or less people kind of know a little bit about the scale and everything else, but what it really means is a concentration, specifically of how many hydrogen ions or protons are present inside any liquid. For us, it's inside a living thing. And most people talk about this as being a measurement of acidity or alkalinity in a pH scale that goes from about 1 through 14, in which the middle of that range is considered to be neutral, below it is considered to be acidic, and above it is considered to be alkaline or basic. Important things to notice here is the fact that most living things, not necessarily all, but most of them, usually function around that kind of neutral range or that middle range. And so when we talk about disease specifically, we'll get to kind of look at the human body and see that different parts of our body function at different pHs, and whenever microorganisms kind of invade, they'll disrupt that pH as a way of causing disease. So that's really why this subject of ions, and specifically hydrogen ions, are important for us in terms of the calculation of pH. So it's kind of important to know how to calculate this too, and there's a basic formula, more or less, that everybody learns about. Here we're not going to kind of spend a lot of time behind it. All we want to know is more or less how to measure the actual amount of hydrogen ions and the pH number itself. So if you recall any textbook, it's usually about calculating moles in terms of how many grams of an element is based on their atomic weight. And so usually people can extrapolate that using a periodic table. Here we're not going to go that complex. The idea is more or less to estimate specifically concentration. Now, if we want to do that, what we'll do is it'll allow us to calculate more or less how to balance the amounts of ions that are present by keeping them stable, and that's what we call a buffer. So these are substances that allow us to maintain pH inside a living system. So the most common example that people learn about is carbonic acid, which normally is present in your blood, which again, roughly around pH 7, and anytime you challenge that pH, carbonic acid kind of kicks in and prevents your blood from becoming too acid or too alkaline and ultimately killing you.
So all this pH becomes very important in healthcare, right? So when your body's retaining too much acid, your kidneys will actually start to produce the bicarbonate to combat that and reach, like the phrase that we hear all the time in anatomy is the homeostasis, right? Everything should be level, working as it should, because if things get too acidic, that starts causing negative issues, you know?
Right, and so blood is the most sensitive of them all, usually kind of distribute all over your body, but anything from kidneys, like you said, your liver also is more or less kind of very alkaline, and so people usually challenge that depending on their diet, and they kind of change the pH. Yep. Same thing, so the idea is kind of important to understand that pH needs to be usually stable, roughly around 7 in most places for most living things, but we'll definitely see some instances in which that's not necessarily accurate. So it's kind of important to know pH in terms of the standpoint of what ions are and ionic bonds, and again, acids and bases. This is why we learned that subject.
All right, so let's get into covalent bonds.
Right, so we just saw ionic ones. Again, so an ionic bond is based on this idea of two opposite charges attracting. Here the covalent bond's no longer about electricity. It's more or less about sharing. So instead of atoms in this case, or ions, kind of going back and forth between electricity, here it's more of a sharing situation. And so usually we still get those pairs that we were looking into, but more or less what we're trying to do is two electrons just come together, and instead of one going to one side and one to another, they kind of stay in the same area, keeping the element close by. So the two elements end up kind of sharing that pair of electrons, and usually this becomes far more stable. And so covalent bonds, as opposed to ionic bonds, are fairly strong, and opposite to our ionic bonds, that they don't usually dissociate in water. So these do not create electrolytes. One of the common examples that we have listed here is glucose, an important substance that we'll talk about energy a little bit later. But this one, when you remove it and you place it into water, it actually stays intact. It does not conduct electricity. It does not break down in water. It does not separate out into carbon, hydrogen, and oxygen. It remains exactly as glucose, as opposed to something like table salt that we were talking about a little bit earlier. Now, there are several versions of the covalent bonds, and they're all based on just how many you can form and their length and strength. And these are classically known as single, double, and triple bonds. More or less, people have seen these in textbooks and everything else too. But more or less, the concept behind them is, how do you distribute a single bond, which is pretty much one pair of electrons going. We talked about methane earlier. This is an example of the chemical formula showing that, the structural formula of it. And you see multiple single bonds. We can have double bonds in which now two pairs of electrons are being shared. And we can have triple bonds in which now three pairs of electrons are being shared. And usually these are the strongest versions of them all, as more electrons are holding two elements together, in this case, carbon. So we'll kind of learn about our covalent bonds as the stronger version of interactions between elements and holding them together. So to kind of visualize this, it's important to see that those outer electrons we were talking about before, they're not going to one side or the other, they're being shared. And again, usually by pairs. So the example that we have on the top left, hydrogen having one pair of electrons being shared, so it has one single bond in there. On the right hand side, you have oxygen on the top, and so here you have two pairs of electrons being shared, and you actually see it as a form of a double bond for oxygen gas. And then you see the variations of something like water versus methane that we saw a little bit earlier too. So just to make sure we clarify the differences between covalent and ionic bonds, one is significantly stronger than the other, covalence being the strongest. Ionic bonds are based on opposite charges, whereas covalent are based on sharing those electrons. Biggest difference is that our ionic bonds will always dissociate inside water, whereas covalent ones will not. And the other side property we have for ionic ones is that those are the ones that lead to acids, bases, and us understanding pH. So those are the two main ones.
All right, so getting into hydrogen bonds, these tend to be some of the weaker bonds, right? They're like just very slight variations between atoms.
So what's happening between those atoms is here, now we're not talking about atoms anymore. What we were talking about before is taking one carbon, one hydrogen, one oxygen, and so on and so forth, and bringing them together. Here it's no longer inside a molecule. Here it's outside the molecule itself. Hydrogen bonds are these type of interactions that form when there's partial kind of almost ionic charges occurring between certain atoms, meaning kind of many positive and many negative charges. Now, hydrogen bonds, again, are only holding molecules together, not within a molecule itself. And like we had mentioned earlier, these are typically fairly weak. But as opposed to an ionic bond that is also weak and breaks down in water, or a covalent bond that happens to be very, very stable by sharing electrons, hydrogen bonds work by mass action. In other words, one of them by themselves doesn't really do much. But millions and millions of these actually end up creating one of the strongest forces that we have, and this is what pretty much maintains most life. One of the easiest ways to kind of exemplify that is by reminding you, anytime you try to boil any liquid, most liquids kind of go away fairly fast if you kind of set them on fire. But water, you'll realize that you have to put a lot of energy, a lot of time, a lot of heat before it barely starts kind of breaking down from a liquid into more of a gaseous form too. And that's because of the amount of it. It's not necessarily that each hydrogen bond is weak itself, it's the millions and millions and possibly even billions of these coming together. So mass action holds them together. So a visual way of kind of showing this is, again, these are partial charges. They're not full ions, but they're not covalent in any way. These are just kind of small pieces of the atoms within a molecule kind of becoming a little bit more charged. And because they have partial charges, then they'll start having a little bit of that attraction too. Water, more than anything else, which is what you're seeing on the right-hand side, shows partial negative and partial positive charges. And so these weak forces, each one of them starts kind of surrounding each other and in mass create one of the strongest forces that we know in terms of living systems, which are your hydrogen bonds. So to summarize this, we'll have all three bonds, ionic, covalent, and hydrogen. And the difference here is that two of these, the ionic and the covalent ones, are what we call intramolecular bonds, in this case meaning within a molecule, so inside water, inside methane, and so on and so forth. Whereas the hydrogen bonds are intermolecular bonds, meaning amongst molecules or between molecules, and that means it's one molecule of water interacting with another molecule of water, or one molecule of methane interacting with another one of methane. The idea is between molecules, not inside one of them themselves. Now, just kind of some reminders of this topic is some of the common mistakes that people make in exams is to remember the differences between atomic numbers, which means the number of protons, and the difference to atomic weight, which means the number of protons and neutrons. To remind also people as we're reading periodic tables is that protons and electrons are usually the exact same number, but the neutrons usually can change a little bit. And also that radioactivity and isotopes specifically, it's the number of neutrons that change, not the number of protons and electrons. And lastly, pH is another common misconception of we're looking in terms of ions, specifically hydrogen ones, not anything else. So those are kind of the big kind of mistakes that people make in the exam. So remind people to study that a little bit more.
All right, so moving into organic chemistry, that's mainly based on carbon, right? We're all carbon-based life forms.
Right, so you've heard that term before, so what we're going to do with this is kind of explain what does that actually mean. So the idea behind organic chemistry is studying carbon. And the reason behind it is because carbon happens to be very unique in the fact that its electrons that it has on the outermost shell, all four of them are kind of equally powered and symmetrical. So that means that all four of those can form these really, really neat arrangements, typically long chains of them or even rings, and they all form covalent bonds. So that means that these chains or rings are fairly stable and fairly strong. So that's what really typically allows life. Now these carbon bonds, again, what we're going to be interested in of them is what you can add to them. In other words, once the chains or rings form, what can we do with them specifically? For that, we start talking about what we call a carbon skeleton, meaning that most of the time, the most basic carbon chain, which happens to be a 1, that's not really a chain, has a specific arrangement in space. And so three-dimensionally speaking, that kind of takes this so-called tetrahedral arrangement. It looks kind of like a pyramid in space. And that more or less ends up looking kind of like a couple of the hydrogens kind of sticking towards you, a couple of them going towards the back, and it gives them more this 3D arrangement of itself. But that's just one of them. We want to see them in chains or rings. And as you start getting them longer and longer, the three-dimensional arrangement of them gets a little bit more complex, and it kind of looks a little bit more challenging or daunting to kind of memorize that. So thankfully, organic chemists have had an agreement on how to kind of summarize all of that by more or less removing all the hydrogens and assuming that they're there, and even removing the letter C from carbon in there, and just kind of simplifying it into a skeleton. And so that's usually what we use in this class kind of as a way to draw out these compounds or these organic compounds. And all it represents is that at each corner there's a carbon, and at each carbon there should be the correct amount of hydrogens, which typically means for carbon it's a grand total of 4 bonds maximum. Now again, like I said, carbon forms the skeleton itself. That's the most basic component of it, but it doesn't really do much by itself. Instead, there are a couple of other biological important elements that add to it. So here we have things like sulfur, phosphorus, oxygen, and nitrogen, and we've already kind of covered hydrogen. Now why they are a big deal is because each of these elements have a unique bonding ability. So they usually end up bonding, or creating bonds I should say, that are kind of either odd or even and allow these really intricate kind of arrangements. Now once they do their thing, they actually become very biologically kind of imperative in terms of what they can create. So in terms of, for example, carbon, it's base of everything. All of life uses carbon, and so same thing applies to hydrogen. But things like nitrogen happens to be very rich in both your genetics, so DNA and RNA, as well as the composition of proteins. Oxygen, on the other hand, is virtually found in most of our living substances. Phosphorus, for example, is highly rich in our genetics and DNA and RNA. And then lastly, sulfur is found typically in proteins. So these are what we call the 6 biologically important elements of life itself. Now, explaining again that carbon does not exist in a vacuum for us, it's all about life, it's kind of important to know what we can add to carbon, and that's the concept of a functional group. Functional groups normally are things that are added to the carbon skeleton, so that means that the carbon skeleton again does not exist in a vacuum, and so these are substances, molecules, compounds that get bonded to the skeleton and usually change the way that the carbon chain functions, hence the term functional group. It usually alters the behavior of the carbon chain. So there are several functional groups out there. We're only going to highlight a few of them, reminding you again that the functional groups don't exist on their own. They're usually added. For example, the so-called hydroxyl group or alcohol functional group, that little OH that you're seeing there, when it's added to a carbon chain or a ring, it makes that particular substance alcoholic, meaning it has the property of alcohols. Usually they become either flammable or they become reactive or something like that. Methyl groups, on the other hand, adding another carbon extension to them. Amino groups usually introduce nitrogen to those fields. Carboxylic acid groups would end up introducing extra oxygens and hydrogens, which one of them comes in the form of an alcohol, and they create certain hydrogen bond properties. Phosphate groups that we're going to be dealing with a lot, present in our DNA and RNA, for example, give electrical character and certain behaviors that allow the chains to get longer and longer. We have carbonyl groups that come in two forms called aldehydes and ketones, which both of these have certain reactive properties also to preserve materials. And the last one that we have are ester groups, which is an extension of an oxygen using a carbon usually, and again helps us form longer versions of the chains. Now, it's not really important to know the structures and details just to be able to identify them. So in other words, can you see what's contained within them? And then as we go throughout the classes, we'll actually see what each one of them brings uniquely. So kind of like what we talked about the chemical reactions, we'll see these as we go. Now, what we're ultimately going to form with these is what we call complex organic molecules by combining these functional groups with carbon and then making or building larger items, larger substances. And so we'll start with the most basic of them. These are called monomers and polymers. What it means here is that these are repetitive units or similar units that usually end up existing in chains. Monomers in this case being the basic subunit of them, and polymers being the entire complex put together. And what we learn about these is that we can build polymers from monomers by using the two chemical reactions that we talked about a little bit earlier during the chemistry portion, called dehydration synthesis and hydrolysis. So how do we form these? One of the easiest concepts is how to build a polymer, and the idea is kind of like doing an addition. You take one monomer and another monomer and you build it into a two monomer system, also known as a dimer. Now there's something curious about this, is that in order to attach them together, usually this involves the removal of an alcohol group, highlighted in red, and a little bit of hydrogen. What's really cool about this is that once you remove those, they end up kind of joining together and forming water. And this is what brings us to the name of this substance, or this reaction. That means this is called a dehydration synthesis. Why? Because you're technically synthesizing a polymer by putting two monomers together, but you're also removing water from the system, and hence the term dehydration. So a dehydration synthesis reaction is all about building polymers by taking two monomers or more, obviously, and removing some water for them. Directly opposite to this, we have the breakdown of these polymers. In other words, you can take a larger substance and force water into it and break it down into more simpler monomers. And so this one, we actually use a little bit of the same terminology. We use the term water, the term hydro, but then we also use the term lysis, meaning breaking. And so the reaction here is called a hydrolysis, as a way to force water into the system and separate something complex into something smaller.
All right, so kind of a quick recap. You were saying that monomers can be built up into polymers, so that builds up these large chains, and we call those macromolecules, right? Those are like the bigger pieces. And people who are into, I know a lot of people who are into fitness and nutrition know a lot about these. We're talking about carbohydrates, lipids, proteins, things that our bodies have evolved to utilize, right? There's like building blocks within.
Correct. And so that's kind of why we address this in this subject. Again, not necessarily to the deeper organic chemistry side. It's more or less what we build with them and how they keep us alive. So we already mentioned some of them. And so the grand total of the macromolecules that we're interested in are those carbs that you just mentioned, the lipids, proteins, and then typically the one that most people forget is the nucleic acids. But we'll explain them all. So what we want to do is go one by one, kind of highlighting why they're important, how they are built, and more or less their impact in terms of life. So we can start with carbohydrates, and here the term carbohydrate indicating it's largely composed of carbon, hydrogen, and oxygen, which usually kind of represents water, hence the term carbohydrate, but this comes in long chains. So that N right there is kind of telling us that there's multiple of these, not just one. Now there's two ways to kind of analyze this, is in its simple form called a monomer, so the basic subunit, and we call that one is referred to as a monosaccharide, or often called a simple sugar is another term for it. And when we build these up into long chains, like we were talking about before, these become polymers. Now what will normally happen is these polymers kind of can exist in chains or in rings of these, and these polymers, called also polysaccharides or complex sugars, play a massive role in terms of life. In other words, why it's important to do these is because they can form these very, very long chains of these rings of carbohydrates, and those can function in many different things like storage. For example, we can hold energy inside of them. So for us in animals, glycogen happens to be how we store our sugars for later use, but plants also use it to store their energy in terms of starch. Another good use about these long chains is that they're fairly rigid and kind of strong. So the fiber that you find in plants or the bark that you find in trees all kind of use the same thing as a way to use it in structure. Now, some things that get forgotten about carbohydrates, they also play a major role in producing proteins by modifying them. So after we create proteins, which we'll see in a few moments, sugars or carbohydrates are normally added to create or modify those proteins and give them additional functions. And then the last one that people shouldn't forget about in terms of carbohydrates is that they are the main component of our DNA and RNA. So we have more or less carbohydrates or sugars in all our DNA and RNA. So carbohydrates as a macromolecule play an essential role in terms of energy, proteins, and our own heredity.
All right, so talking about lipids, these are long chains again, and we'll talk about in a second. Some of them are saturated and unsaturated with hydrogen, correct?
Right, and so that's kind of an interesting property to talk about. So that's one of their definitions, right? These are long chains, like you mentioned, but usually they carry massive amounts of hydrogen. The term saturated happens to be one of those terms. But the other thing is that because they have lots of hydrogen, meaning that they have very low amounts of the other functional groups, is that they don't really mix with water very well. Something that most people kind of assume, this whole idea of oil doesn't mix very well with water, right? So they typically carry very small amounts of oxygen and any other of the other elements. So they're usually lots of carbon, lots of hydrogen. Now we are going to cover three variations behind them. And so we're going to see them one by one. The first group called the triglycerides, second group called the phospholipids, and the third one calling the sterols. All more or less related because these are all three types of lipids and all have some sort of biological importance. First one being here are triglycerides. Typically, commonly, people abbreviate them as Trigs. Here it comes with two pieces, again, understanding what they're made out of. One of them, they're composed of a small sugar called glycerol. This is the substance you're seeing on the right-hand side. 3-unit carbohydrate right there. And the larger part of a triglyceride is what we call a fatty acid. And these are the terms you were bringing about earlier. Is that they come with very, very long chains of carbons with very, very little oxygen or anything else. It's mostly hydrogen. And based on the amount of hydrogen that they carry, they can be referred to as saturated fatty acids or unsaturated fatty acids. And the differences are quite obvious. When they're full of hydrogens, not carbons in this case, so they have the maximum amount of hydrogens present amongst each carbon, this is referred to a saturated fatty acid. You'll notice again, virtually everything is covered in hydrogens with one tiny amount of oxygen present. However, you can also have unsaturated fatty acids, and those are the ones that are missing hydrogens, and they usually create double bonds, and they actually introduce changes into the structure of the lipid.
So something interesting about the saturated fatty acids, because of the way that they're shaped, they're so uniform, they tend to kind of stick together, right? And so when you see fats that tend to be like more solid, like butter, those are the saturated fats. And then the unsaturated usually have like kinks, and they don't fit together as well. They're not so, Lego like, so they tend to be more like oils, right?
Correct. And so based on their structure, again, they dictate more or less their solid or liquid states. And so that's kind of important to know when you're consuming these, is you get to know what you're consuming based on saturated or unsaturated just simply by their structure. Now, so when we put them together, we take saturated and unsaturated fatty acids attached to this glycerol unit, and since we have 3 of them, typically, this is the term triglyceride. So we take that glycerol unit and 3 fatty acids, and they can be combined. It can be all typically saturated, all typically unsaturated, but most of the time, about 90% of the cases, these are combined. A couple of saturated, a couple of unsaturated, and so they give unique properties to these triglycerides. Now, why are they important in terms of biology? Specifically, their function. Their function specifically is important in terms of retaining heat, a concept known as insulation, but also because of their large kind of chain nature, they also function in what we call a cushioning effect. In this case, the idea of not only retaining heat, but also being able to absorb shock. So this helps us in protecting our tissues more than anything else. And lastly, most of the time, these lipids also can contribute a small amount of energy depending on how we break them down. So our very first type of lipid, our triglyceride, plays a role in, again, heat retention, cushioning, and a way of providing a small amount of energy. Right after that, then we introduce the concept of a phospholipid, which is virtually the same thing as our triglyceride earlier. And here what we have is it takes the same concept of a glycerol and some fatty acids with one exception. At the 3rd carbon of the glycerol, now it's missing one of those fatty acids, and instead it replaces it with a phosphate functional group. And so you can see the 2 long chains there, and then you have one of them missing, and instead of it you have a phosphate functional group attached to it. Now that phosphate usually changes its properties by making it more electric, and so it gives it a more ionic property behind it too, which will come into importance later. Now, why do we want to learn about phospholipids? Is because primarily, more than anything else, since we've been talking about cells, all cells are made up of phospholipids. Their main component in terms of its structure, in terms of its shape, in terms of more or less that cellular kind of bubble or sphere that most cells take, is the phospholipids. So this is a critical component in the building of all life. A unique other property that phospholipids have is because of that phosphate is that they can help you kind of break down and separate other lipids, the process known as emulsification. So this is kind of a side property that it has, but more or less the reason why we learn about phospholipids is because of the building of all cells. Now the last one that we're going to deal with in terms of lipids are our sterols. Another term that probably you've heard behind it is called a steroid. It means the same thing. And these are lipids that instead of being long chains, now they come in rings. So they have this kind of pattern of 4, 5, 6, even 7 carbon rings. And these kind of become interlocking, so they're interacting with each other. And sometimes you get 2, 3, 4, 5 of these rings also interacting. Now, what's unique about these guys is, again, they usually have a tendency of sticking together, much like your oils and butters too. But their main component that everybody refers to as a sterol is cholesterol. But here, cholesterol is learned more as a structural compound, meaning it helps build also our cells. So it's a integral component of most cells too, most cell membranes. Without cholesterol, there cannot be cellular structure. Another unique component behind it, or function, is that they contribute to the breakdown of lipids also, kind of similar to phospholipids. And these are a component of bile acids. So when you're undergoing digestion or you're trying to break things down, sterols have a tendency of helping you break things down too. And then last but not least, one of more of an animal or mammalian component behind this is that they contribute to hormones. And so most of the steroid hormones that we're familiar with, things like testosterone and estrogen, follow this same structure too. So they also have a signaling component behind them. So this is why we learn about sterols. So these are our 3 lipids. We have our triglycerides, we have our phospholipids, and then we have our sterols as part of those lipid macromolecules that we have.
So following on with macromolecules, let's talk about complex macromolecules. So we've got, this is when you get into, you mentioned DNA and proteins and things like that, that I know they actually like can carry information, things like that, that get a little more complex, right?
So the complexity that you're referring to here has to do with what they do, as opposed to what we just saw, carbs and lipids. But now, these ones not only are complex in terms of their functions, but also size. So there's two big stories behind this. Our proteins, in terms of what they can do, like you said, they become very complex simply because of their size, but also how many things they can perform. And pretty much we know that proteins virtually do everything in a living system. In other words, any type of function you can assume in a living thing, it's typically being done by a protein. And so that can be anything from building things, signaling, transporting, motion, digestion, and so on and so forth. Proteins do all of these jobs. Whereas nucleic acids, well, their term usually kind of gives us a basic information behind it. This is our genetics. In other words, these DNA and RNA molecules are all about transfer of information and more or less our heredity. So these two molecules, the reason, again, like you said, were complex, because their size, they're much, much larger than the previous two, but also they perform much bigger roles in the living systems. So let's kind of take a look at them very briefly. Our proteins, usually kind of learning what their basic unit is, a monomer, so the smallest subunit of them are referred to as amino acids, probably a term that most people have heard at least in the past. And these amino acids are composed of a very tiny carbon skeleton, usually 1 or 2 carbons, and they possess 2 functional groups, an amino group and a carboxylic acid group, and usually what we call a variable group or an R group that is more or less giving it an additional function to it. And so this is what we're seeing on the right hand side, kind of color coded. And that R group or that variable group usually can come in up to 20 different variations, which is why we end up talking about 20 amino acids a little bit later. Now, amongst that, though, is that that simple unit can be built into polymers. And so the same idea, dehydration synthesis, will remove a little bit of water and connect those 2 functional groups to build what we call a polypeptide chain, or what most people assume is called a protein. Now, not forget that our carbohydrates that we talked about, one of their main functions is also to modify these proteins. So typically speaking, once your proteins are formed or your polypeptides are formed, sugars or carbs are added to them to modify them. So what normally happens here is you'll take 2 monomers of these amino acids and you'll remove some water, and that will kind of bring them together. Except that this usually happens in such a long version of it that instead of taking 1, 2, or 3, or 5, or 20 of these end up being in the hundreds to thousands of amino acids put into what ultimately forms polypeptide chains. So again, going back to its complexity and its size, lots of different functions in a very large macromolecule. So one of the key features to understand here about proteins is what they do. And like I said before, they can do a lot of things, but it's all down to what their shape is. One of the more important things about proteins and their functions is more or less their 3-dimensional structure. Once that structure is lost, they stop behaving the right way. This is a process known as denaturation. And so once we get into the genetics class, we'll talk a little bit about everything else associated. So the short story here is what they do is virtually everything. They perform every single or almost every single function in most living things. Well, this is why their importance is critical in microbiology. Now going to the last of the macromolecules again, size and complexity in terms of what they do. Here we have nucleic acids, and their monomer, their smallest subunit, is known as a nucleotide. Their nucleotide is also fairly small in terms of being 1 sugar. Remember, sugars or carbohydrates are a main component of nucleotides. And they come in several different variations of what we call riboses and deoxyriboses. And attached to them, we usually have a phosphate group, functional group attached to it, and something called a nitrogenous base, a more complex functional group to them. And so what we normally have here is that that nitrogenous base can come in about 5 different variations. And those 5 different variations will start forming these long chains. What we call polymers of them are called nucleic acid chains. Here what we take then is that that sugar that we're talking about, that carbohydrate, and that phosphate end up undergoing dehydration synthesis, and more or less that little kind kind of sugar and phosphate attachment ends up making larger chains through dehydration and end up forming thousands and thousands, and up to the point that these become millions and billions of chains of phosphates and sugars, and ultimately what most of us know as our DNA and our RNA. And so here, again, complexity, size, is that amount of sugars and phosphates attached to each other ends up giving an ability to store information. So what you guys know as your genes, your genetics. So ultimately, putting all of those together, you kind of compact them to fit inside cells into what most people know as chromosomes. So that is one of the more important reasons of understanding these complex macromolecules, is that they're kind of usually built upon from the smaller ones, and that they can perform much more complex functions than the previous few. So what are the big deals behind DNA? Well, this is your pretty much sequence of life. This is what are all the instructions or blueprints on how to build everything, and they're usually condensed in your chromosomes. RNA, on the other hand, does come in about a few different flavors, and so mRNA being the more important one of the three, in which this is the one that continues to carry the genetic information. And so the other two are more or less supporting the rRNA and tRNA on how to decode the genetic code that everybody carries. So this is really where all four of the macromolecules that we discussed, carbohydrates, lipids, proteins, and nucleic acids, all kind of give us an idea of how important they are to maintaining life. And that's what we're going to start using in our next set of topics for cells.